So let's start with the first part of kind of reviewing for the base. So I'll highlight things and try to keep things kind of as organized as possible so you guys have a good understanding, particularly as you're going through reviewing. So the first kind of main question that we need to kind of remember is what is biotechnology, or what is the definition of biotechnology? And so that is going to be the use of living organisms or biological systems to create products that improve life or solve problems. So think about insulin. Insulin is going to be the thing that we always come back to. So let's just write living organisms as tools. All right, so ancient biotechnology, or like the old way, used things like yeast, microbes for yogurt, selective breeding. So that's, choosing what's the best cattle, what's the best breed of cattle. So let's say the old way was selective breeding and fermentation. And then the modern way is manipulating DNA in labs. Um, so using genetic engineering, right? So using human insulin to make that product. So the new way is genetic engineering. So biotechnology applies tools from biology, chemistry, physics, and computer science to develop useful products, right? That's the big thing about biotechnology. We are trying to make products that improve life, improve our way of life. So, what are the types of things used in biotechnology? Well, what the first kind of one is going to be enzymes. Remember, enzymes are a type of protein, and you could think of these as like a molecular scissor for DNA. So they help to, so scissors for DNA, and they're used to cut and insert DNA into other organisms that allow for genetic modifications. Tools like chromatography help purify proteins or other substances based on their properties. So, chromatography is really going to be about separating molecules by how they move. Then you've got mechanical tools such as a spectrophotometer, and these measure how much light is absorbed and helping to identify. So, a spectrophotometer is really just a light meter. Depending on what substance is in that solution, um, it can identify about how much light it absorbs or how much light it bounces off. So it's a light meter for molecules. And then polymerase chain reaction, or PCR for short. This is like the photocopy machine for DNA, um, and it allows us to take a very small sample of DNA and grow it into many, many copies. So PCR is like a copy machine. So let's write some tips down here at the bottom. So let's put don't mix up PCR, right, which is polymerase chain reaction, and that amplifies DNA. Right, amplify is just a fancy word for makes copy. Don't mix a PCR with, let's say, restriction enzymes. And remember, restriction enzymes, those are going to be the enzymes that cut DNA, that allow PCR to happen. So let's just put like a yellow star down there, just so you know this is like a little base tip. All right, let's talk about bioethics. So everything in biotechnology raises an ethical issue. Um, Things like being able to change DNA. Um, it's that just because we can, should we? So changing DNA has a lot of power and responsibility. That's right, responsibility. Remember, people fear genetically modified organisms or GMOs because they believe that those products may be harmful to their health. In 1975, scientists got together and began to discuss the rules to safely do genetic engineering. And this is important because the 1970s is when biotechnology really blew up. In Asilomar, that's the location of the conference where they were at. So 1970 is a very important decade in biotechnology. So ethical questions are around cloning, gene therapy, genetically modified foods, using engineered um, viruses. So remember, so let's put our star, our yellow base star for things that we need to remember. So ethics does not equal safety rules. Ethics are about should we do something. And then safety is kind of like how it says. Safety is about how to safely do genetic engineering, etc. All right, who uses biotechnology? Remember, the main driver of biotechnology is going to be the healthcare industry. Um, they use biotechnology to make drugs, to develop new treatments, uh, be able to better diagnose, and to make personalized medicine, right? Remember, personalized medicine is going to be treatment based on a person's individual genetic profile. So a not a one size fit all. The next big industry that uses biotechnology is going to be agriculture, right? So things like soybeans that are genetically modified to resist herbicide, or animals that can produce more milk, that have more yield. They can resist any sort of pests that may bother them, or we can fortify those plants with more nutrition. So think about vitamin A in rice. or bananas with vitamins in it. Um, the next one is going to be the food industry. So the food industry is going to use things to improve fermentation, food processing, or being able to fortify food so that they stay good for longer. And then the next one is going to be environmental biotechnology. So these guys are going to be more concerned with things like how do you clean up pollution, or how do you reduce industrial waste? And that definition, and I think we talk about it in another couple of pages, but just to reiterate, that's going to be bioremediation. So let's put our yellow kind of base tip star. So key thing is going to be to not mix up agricultural biotechnology so which is about crops, and environmental biotechnology, which is going to be about pollution. All right, so let's go to the next page, and feel free to pause the video as we go through if you need a second to write. All right, so we said earlier biotechnology and healthcare. Healthcare is going to be the biggest user of biotechnology. And part of that is going to be the development of drugs. Before you can develop a drug, you have to discover what products, what molecules are effective or can be used to target proteins in your cells or cells of the body. So, drug discovery is going to be discovering new molecules. Once that molecule has been discovered, it then goes into drug development phase or preclinical trials. And there they just improve the molecules and they test them for kind of safety and effectiveness in animals and kind of other um, in vitro situations. So once we go through those two steps, now we enter the three phases of the clinical trials. So clinical trials, these are going to be on people, mostly volunteers. And here they're going to check for three things. They're going to check for safety, right? They're going to check for dosage, and they're going to confirm that this drug will work. So success of the drug. And so I'm going to come down here to this picture real fast, right? And it's going to be really important that you know what happens in each of the phases of the clinical trials. So phase one is safety, and that's going to have the smallest amount of people. Phase two is going to see if that drug is effective against the treatment. And that's going to have the second most amount of people. And then the third phase is going to have the most. And once again, this is just going to check for large scale effectiveness. So it's really important that you know what happens in all three steps. And then we've got personalized medicine, right? So this is going to be, instead of that one size fits all, this is going to be medicine made for your individual genetic makeup. So think about Herceptin. Herceptin is that drug for breast cancer, and it's only used in patients that produce this HER2 protein, um, or the marker for that. So if they have that marker, then they will be prescribed Herceptin. Otherwise, they would not be. All right, so just maybe I'll just write up here biotechnology and healthcare largest user. All right, so now let's talk about biotechnology in agriculture. So this is going to be genetically modified crops, and if you're curious as to what GM stands for, let's just write it to make sure that we're clear. It's going to be genetically modified. So these are going to be things like corn, these are going to be things like rice. They're modified to resist pests that may eat the crops. They can tolerate herbicides, so the farmers can lay down herbicide, kill weeds, but not kill their crops. Or they can have extra nutrients in it, like vitamin A in golden rice, which they sold to developing countries. Um, Dairy cows can be treated with a growth hormone. That growth hormone is going to be recombinant bovine somatotropin. This is just an example. And then let's just write down over here. So the word soma means body, right? And tropin means to grow. So a growth hormone that makes cows produce more milk, which can be controversial. Bacteria in food that they use to resist viruses and produce higher yields of products like yogurt and cheese. And then if we come up here to the top, so plants are being developed to produce medicines or vaccines, making healthcare cheaper and more accessible in poorer regions. So let's just do a base reminder here. So, not all GMOs are bad. There's definitely a lot of good for them, good uses for them. So not all GMOs are bad. So all right. So industrial and an environmental biotech. So these are going to be things like enzymes. Once again, so enzymes keeps coming up. Right, so enzymes use harsh chemicals in textiles, which they soften fabric without damaging machines. So think about like softening jeans, like stonewashed jeans used to be with stones in the machine, and they would wash the jeans to get that fade. But however, now we can use an enzyme to produce the same thing and not damage those machines. Cold water laundry detergents, these use enzymes um, to be able to wash your laundry, wash your clean clothes without having to use hot water. And then things like bioremediation, which they use to clean up things after oil spills. So, like the BP oil spill in the Gulf or other pollutants in the soil. And biofuels, these are going to be more environmentally friendly fuel sources that are renewable and cleaner. So let's just do kind of base reminder, key thing to look for on the test. So if something says bioremediation, think that cleans waste, and biofuel makes energy. All right, kind of moving on, lots of careers in biotechnology, right? So, not just for technicians, but you also need people to sell the products. You need to make sure that the products are safe and that you are ensuring that everyone's getting the same product. Biotech is a very rapidly growing field, and just about any industry you want to go into, manufacturing, healthcare, the environment, everyone is using biotechnology in some form or another. You can begin as a lab assistant, as someone who just helps out. So, like doing things like the BACE test, you can move on to an associate's degree. And then as you get higher up, they may require you to have a PhD. So, kind of the tip to remember from biotech careers is going to be not all biotech jobs require a PhD. All right, so now let's go to the next part. So now this is going to be about lab safety, right? So this is going to be about safe lab operations, good record keeping, um, using PPE. So let's just kind of highlight PPE, right? That's personal protective equipment. All right, so just general safety guidelines, gloves, lab coats, eye protection. This chart of hazard symbols, I would highly, highly know these signs because these signs will come up on the test, and these are easy points that you can get. So know these hazard signs. If you are working with something that needs to require a positive or negative airflow, you're going to use a fume hood. So any sort of aerosol. Labs are going to be considered between BSL 1 and BSL 4, 1 being the safest and BSL 4 being the most dangerous. Think about some crazy virus like Ebola. Most school labs are not going to be handling anything crazy, so these are going to be BSL 1. Waste has to be separated or segregated and disposed of using an autoclave. When in doubt, autoclave out. So that's the gold standard is the autoclave. And if you don't have that, 10% bleach solution as needed. All right, so now let's go up here. Let's talk about maintaining the lab notebook. So the lab notebook, key thing about that is going to be you're going to want to record every single thing you do in your lab. The title of the lab, the purpose, methods, materials, results, conclusion. Big thing about lab notebooks is that they have to be in black ink. And then errors must always be crossed out with a single line. Your lab notebook should include tables, sketches, figures, calculations. Entries should be complete, legible, and dated. And just remember that lab notebooks are legal documents, and legal documents are necessary to establish patent. All right, so here's just an example of what the lab notebook should look like. All right, to measure substances, right, a micropipette, what's commonly used, this is going to be for 1 microliter to 1 milliliter. The graduated cylinder has moderate accuracy, and just remember you're going to read this at the bottom of the meniscus. So if this is your graduated cylinder here, right, it's got all these lines on it, and here's your liquid, kind of down here. So you're going to want to measure at the bottom of the meniscus. Pipettes, so not a micropipette. These are going to have one fixed accurate volume. All right, and then using to measure mass, so this is going to be to use weight. Whenever you're measuring something, always weigh with weight boats or weighing paper. So just to have something between the scale itself and whatever you're measuring. Alcohol thermometer and digital thermometers measure in Celsius. Remember, Celsius is going to be the temperature that we use. pH meters, you're going to want to calibrate these with buffers that are 4, 7, and 10. All right, talking about the autoclave, this is going to be important. So the autoclave is going to be at 121 degrees Celsius and 15 to 20 psi. So that is, and remember this, and this is going to be for sterilizing, so it kills everything. If you don't have that, your next best solution is to use a bleach, a 10% bleach solution. All right, so now let's come up here. Let's talk about significant figures. Significant figures reflect instrument precision. So when you're doing addition or subtraction, you round to the least number of decimal places. If you're multiplying or dividing, limit answer to the fewest significant figures. And so you would just look to see how many significant figures you have in the question and answer to that. So answer, and let's do our little base star here. All right, so scientific notation, it's going to be the next thing. Um, this is just going to be used to shrink or expand big numbers. So instead of writing a bunch of trailing zeros, you would just put it into scientific notation. All right, metric prefixes. So these are just going to be to shift the decimal one way or another. If you're not familiar with the metric chart, I would probably know this as well. Um, Really, you're going to be going a lot of times from milliliters to liters. So just about moving that definition. All right, preparing solutions. I'm not going to spend too much time on the preparing solutions because I think I'll just make another video over the math. But just a couple of kind of formulas you should know, right, is going to be mass over volume, so M over V, or V over V for volume over volume. For dilutions, you're going to use the dilution formula, which is going to be concentration 1 times volume of 1, so C1 V1 equals C2 V2. And so this is really whenever you see the word stock, right? And then you're just going to use this formula. So X concentration solutions. Then there is going to be molarity, so that's moles per liter. That's another important one to know. And the final one is that on just about every BACE test that I have seen, there has always been a microscope that you will have to label. And once again, I think these are really easy points to kind of get through. So the lens you look at, that's going to be the ocular lens. The lens that looks at the material, that's going to be the objective lens. Where your sample sits, that's going to be the stage. You have a diaphragm that lets in and reduces amount of light. And then at the bottom, you have the light source. You also have a couple of adjustments that you can make. So you have coarse versus fine adjustments or fine focus. So coarse is going to be for big movements, and fine adjustment is going to be just making those small, tiny adjustments. That's right, tiny adjustments. All right, so that kind of concludes the what is biotechnology phase, or kind of examples. Um, the big thing is, especially as you're getting ready to take the test, it's just going to be to keep an eye out for some of the key vocabulary that hopefully I have blocked off and kind of explained a little bit. I will make another video for the math so we can be really comfortable with the math as we go through. Definitely know the hazards, know the picture of the microscope. I think those are going to be some easy points you can pick up. Another place where you're definitely going to see questions so that you could pick up easy points is going to be about the clinical trials. Just know the 3 phases of clinical trials and that's happening in humans. And all right, so just be on the lookout for the next video. Thanks, guys.